20-minute drive up. Great to be so local. Been working with David for the past 6 years now, which is crazy to say that. But David bought the Tyto cell sorter back in 2020, and actually it got delivered a week before everything shut down for COVID. So that's always an interesting way to start off, you know, a new instrument. But it's been a really successful instrument. We'll talk about that later on. But a big focus of our talk today is going to be about sample prep and kind of key practices and ways to optimize your sample upstream of your sequencing, right? Because that is a huge, huge factor, right? We like to say garbage in is going to be garbage out. So we want to make sure that what you're putting in is going to yield the best results. So you're not wasting your time, you're not wasting your money, and you're excited about your results. So the team we have here, obviously I'm the flow cytometry specialist, but for us right here, he's a flow application scientist. So he's your technical go-to for anything for flow cytometry. So if you guys have ever used the Tyto, if you have questions about the Tyto, if you want optimization techniques, panel building, he's the go-to guy for that. He's helped a lot of customers on campus here. And then we have Morgan Little, who is your technical sales consultant. So she handles the entire research product portfolio, which is going to be a main focus for our sample prep portion. So let's get started. And again, feel free, if you guys have questions in the middle of the presentation, we're more than happy to answer them as they come. And again, you guys, we can send all of our information out, but we're all local in Chicago, which is really great for you guys. It's really quick and easy for us to get here. I like to start every kind of presentation just with a quick little mission statement from Miltenyi Biotech, because I don't know if you guys are familiar with kind of our company, where we come from. We're actually celebrating 35 years, which is crazy. And Stefan Miltenyi is actually still running our company, the company's named after, but really our mission is ingrained in helping the research and moving science forward, and elevating just the human condition. So we do that through cell and gene therapy. We do that through our manufacturing of our, you know, our technologies, but we're always coming out with new things. And that's all based off where the market's going and how science is advancing. And so we really appreciate, obviously, our customers, but also input from you guys on kind of how to move that forward. Just some quick facts. Again, something that I like to highlight here is that about a quarter of our commercial workforce is actually in R&D, which is, I think, a really powerful statement, is that we have a lot of scientists at Miltenyi. I would say 90% of the people working at Miltenyi have a scientific background. So that's really powerful. Obviously, the number of patients we've treated, and that number has substantially increased. But we're really here to kind of meet you where you're at in your workflow. And then again, a huge thing that we're going to focus on is start smart, right? So how are you starting off your kind of applications, right? Because that's going to be a huge determinant of what those downstream results are going to be, right? So that's where Morgan's going to come into to kind of talk about our full sample prep portfolio, which is really going to help elevate your downstream results. And we'll kind of walk through all those things, and then I'll kind of talk quickly about the Tyto, and then we'll pass it off to 10X. So I'll pass it off to Morgan.
Thank you, Allie. Hi, good morning, everyone. So what we're going to get started on here today is talking about getting good good quality single cell sequencing data. And the way that we're going to do that is by making sure that we have samples that are free of debris, that our viability is really high. So we want like 80 to 90%, depending on, you know, what assay you're doing, and that they're truly single cells. These can be difficult if you are not doing, if you're not careful with how you're processing the samples. We also are interested in making sure that it's fast. So that's going to help you boost viability, that we're not altering any of the frequencies of the cells in the process, so that our gene expression pattern and our cellular expression are consistent with the in vivo situation. And so we have products that are supported from whether you're starting with blood, or if you're going with PBMCs, or maybe you're starting with tissues. You'll want to make sure that you have a sample that is as close to the situation in the starting material as possible, using a really nice workflow. So we have here the tissue storage solution, tissue dissociation, blood products, and sample cleanup. So whether you're starting with tissue or blood products, or maybe you're starting with a cell from cell lines, you'll want to go into sample cleanup, and then do some sort of target cell isolation, and then followed by cell analysis and single cell analysis. So for cell preparation, I know that like when I was in the lab working with animals, a lot of times I would have multiple or cohorts of multiple sizes, different animal or organs that I was going to be preparing from each animal. And I was always worried about time spent on ice and sort of biasing the sample between the mouse 1 and mouse 20. So the solution to that is this long-term tissue storage. We highly recommend using fresh tissue for all of our assays. I know a lot of people will get into freezing their tissue to get around this time on ice bias, but there's limitations to that, right? So if you do snap freezing of tissue, a lot of times you're limited to nuclei on the other side, and then paraffin embedded or cryoprotectant frozen tissue, you have freezing effects and things like that. So we definitely recommend using fresh tissue, and this will get you up to 48 hours of as close to fresh as possible. So we have some data here. The viability there is in the bar graph. So you can see at both the fresh 24 and 48-hour time points, we see very similar cell viability. And this is at 4 degrees. And then we also looked at some gene expression and we see for stress markers. And at 24 and 48, we have pretty reliable, consistent, comparable to fresh tissue gene expression. So you don't have to worry about the tissue undergoing any sort of cellular changes while it's sitting in this tissue storage solution. And then the next thing we're going to do then is an association. So we like to use enzymes. We have a really nice trifecta of the GentleMax dissociator, which is pictured here, plus our kits and the tubes that all go together to combine mechanical and enzymatic dissociation. And this is going to save you time. And it's also going to give you some hands-free time. So while it's on the instrument, you get that time back. You also have the option to do cold adapted enzymes and also, dissociation without enzymes. We particularly like, usually in most cases, apart from some special cases, heat adapted protocols. And as you can see here, I'll show you some data in a second, but starting with this mouse tumor material and with our kit is going to be this top data, and the bottom data is without the kit, without the heat adapted enzymes. You can see that activated T cells are up to 3 times greater. So our recovery was 3 times greater in those samples that we use the enzymes versus without the enzymes. And that was looking at those TILs that were in those mouse tumors. The kits that we have a variety for both mouse and human. We do have nuclei, so if you are working with frozen samples, we do recommend you do nuclei extraction. And we have a buffer and we have little coolers that actually fit onto our GentleMax dissociator. So you can do everything on the instrument, whether it's hot, room temperature, not hot, but 37, room temperature, or cold adapted. And we also have new FFPE kits. So if you are going to be using FFPE tissue, we have specifically new kits for RNA applications, so RNase-free applications. And that is here. So which kit do you want to use? We have our regular FFPE kit, and then like I mentioned, we have this brand new one for those of you that are looking at RNA profiling. So we can talk offline. There's, I don't know if Ali mentioned, but I covered about 16,000 products, so it's impossible for me to cover everything. But if you have specific questions, we can definitely chat either in the breakout sessions or via email or something. But just to know that we do have, if you have bank tissue, we do have the ability to do your single cell applications from your bank tissue. And then this is that trifecta that I mentioned. So we really like to promote using the GentleMax because it is the most gentle way to get good recovery and a high viability single cell suspensions from your tissues. And that's because it combines the special tubes that provide the mechanical disruption, as well as the enzymes that provide the enzymatic dissociation at the same time. And there are certain cases where you're going to prefer to do warm versus cold. As I mentioned, the nuclei extraction, we do like to have that cold adapted. There's a lot of metabolomic studies that are coming out where they are adding transcriptional inhibitors to the enzymatic cocktail mix to prevent some of those changes that you might see during dissociation. But just keep in mind that any amount of dissociation is going to affect the tissue in a small way. It's just trying to minimize that as much as possible. So there's our workflow again. So now where we are is we have our single cell suspension, and we want to make sure that we get rid of all of the clumps and aggregates, any debris, myelin if you're doing any CNS or PNS work, RBCs, and then any dead cells, just so that you don't waste any money on off-target reads. Or clogging any particular chips or other like systems that you're using. And we have a variety of different products for that as well. So this is an overview. So we have physical strainers at various sizes. I have some samples with me if anybody afterwards wants to pick up a couple samples. But we have filters and strainers that 5, 15, and 50 conical tubes. And then we also have a variety of kits for removing debris, RBC lysis. We have a myelin removal bead, and we do have our dead cell removal bead, which is probably one of our most popular products. This one is going to use our MAX technology. So this is a column-based technology. The beads are going to bind to the either necrotic or apoptotic cells. And so then they're going to bind to the dead cells. Those will stay in the column when they're in the magnet, and then all of your unlabeled, all of your live cells will flow through, and then you can use that for the next step in your process. And we did a couple comparisons of variety of different dead cell removal kits, because there's a lot of options available on the market, right? And our kit is the one there that's highlighted in that pink box. And you can see that when you look at viability, we have the nicest in our hands, the nicest compared to some other popular dead cell removal kits. But if you look at the number of cells, so recovery, because we are removing more of those dead cells and the apoptotic cells, not just the dead cells, we are getting some cell loss. So depending on what your application is, it might not be the absolutely best for your protocol, but we do have 10x recommending this kit, and we do have a lot of people using it in genomic labs across the country. We just launched a new cell storage solution for human tissue. This one is going to be for after you have a single cell solution, you can, similar to the tissue storage solution, where you want to just kind of pause because you have a lot of samples that you're processing, or you don't want to fix them because you're not doing just FACS. Maybe you're doing something you need live cells. We have a storage solution. Maybe you're shipping them to someone else, or someone's shipping them to you. We have something very similar to the tissue storage solution for single cells. So we see really nice recovery up to 72 hours, as well as viability. And then the more complicated graph is looking at the different compositions and to show that they are not affected with the storage. So that is also available. And then just to show some data on how this type of workflow could impact your data, we did a collaboration with 10x where we use the dissociator, the filters, we use an RBC lysis and the dead cell removal microbead, and then we did sequencing. So condition A on the next slide will be just the tissue dissociation and the filtration. B will be with that plus RBC lysis. And C and D will be with the whole workflow, which we recommend. And you can see here that we have, can you see the A, B, and C? Let's see. Well, the filtered only, yeah. So this is the A condition. So the filtered only, you can see that the cell recovery was not as good as and clear and analyzable as the steps where we did the whole workflow, where we had really nice starting material. So we have really nice populations to analyze once we have that single cell data. And then after you have, sometimes after you have your single cell suspension, you want to maybe only look at a subsection. So maybe, you're using brain as a starting material, and maybe you're only interested in the neurons or the glia of some sort. So in those cases, you have a couple options. Ali will talk about the TIDO, which we have here in the flow core, for teasing apart those populations. On your bench or in your biosafety cabinet, though, you have options for magnetic cell separation. And I'm going to show some data here. So this is, again, with brain, but we have this for all different types of cells. Brain is a lab I used to work in, so quite comfortable talking about it. But this is for neuron isolation from adult mouse brain. And we did the brain dissociation with the brain dissociation kit. We actually have 3 different brain dissociation kits, depending on what epitopes you are trying to use later on in your assay. The dissociator, the tubes, we did filtration, we did RBC lysis, and then we also did debris removal solution. And we ran everything on our automated cell separation instrument called the NEO. And this is what that looks like. So you can see without the separation, we have our population is not really distinguishable from the rest of the sample. And then when we actually did do an enrichment, you can see here that we have a really nice segregation of the different populations. And the murine, the mature neurons were enriched. Sorry, this is the one where they are enriched. So this is the sorry, I apologize. So this is without doing the separation, and this is with doing the separation. So we have a very different population of cells to look at, and now a majority of them are those neuronal cell types. And now Ali will talk about.
Are there any questions for Morgan before we kind of branch over into the TIDO real quick? Any sample prep questions? Does anybody have a GenoMax dissociator in their lab or use it? Nice. You guys are the lucky ones. Well, you should be using that. Let me tell you, it's the #1 like requested item on a wish list for labs.
For what? So we have a variety. So what type of cell are you trying to use? We have some kits called multi-tissue dissociation kits, and then we have specific protocols for different types of tissues. And so we would definitely be able to chat with you about it. Maybe I could work with you to develop a protocol, but we don't have a specific standalone protocol. But those multi-tissue dissociation designed, we have 3 of them, and they're designed to be modified to suit the tissue that you're using.
Just to add from Phoenix's perspective as well, this type of approach is really common. So most companies usually have kind of a generalized product that's quite useful across the board, and then there's customization to the specific sample and research question. So reaching out to that more experts is definitely a great step.
Yeah, absolutely. Thank you. Yeah, and one of the nice things about working with 10X and working with MILTENYI is that when you do call for help, you get somebody who has level education, is very, experienced in the type of question that's funneled through somebody who and answer those questions.
Another quick question. So
Yeah.
you guys mentioned the buffers are compatible, the multi-tenant.
Yeah, so I know the EDTA is a problem potentially, right, with the 10X chip. And so if any of our buffers have EDTA, or if you're using any buffers that have EDTA that are from us, there's always going to be a wash step after magnetic separation. And so we would then just do a wash without the EDTA, but they're all labeled whether they have it or not. And then same for the standalone buffers if you're buying them from us, they have it as well.
So which buffer would you recommend? Because I don't know, you look at companies. So back and forth between us and the core facility, we're always concerned about, hey, weather or whatever, self-survival.
Yep. You could put it in that self-storage solution that I mentioned. Then buffer or self-storage solution. So buffer is just, I mean, if it's just buffer, it's usually PBS and then whatever you're adding to PSA potentially.
So for example, I used the title before, right? Title, whatever.
Whatever you want, yeah.
Okay, lines of just transferring it back to the lab and then switch it to the running buffer with the 10X.
For the 10X, what is the buffer that you?
So, to describe that, there's PBS plus 24% BSA is what a lot of our documentation talks about. That range can actually go all the way from 0.02 to 2%. You can go all the way up to 10% of FPS. The thing I always tell people who are nervous about moving between locations is to look for compatible media. So like Max has some buffers that are compatible that we list. There's also stuff like PMAC or PMI. These are made specifically to have, like we were just talking about, more than just buffer. So there's extra nutrients, extra factors that keep your cells alive. The worst thing that could happen is to go through all that work producing the sample and it crashes out in 10 minutes. So these types of buffers are the ones I would push. And I would say it's not necessarily like there's a generalized, hey, this is perfect for your particular application because we have, exact data showing it. More like the suggestions based on experiential learning with customers that we can give you. So it's usually we'll give you one or two options, and the one that fits your logistics and sample best is the one you'll want to pick. Because we want to lean a little bit on your expertise because we don't know as much about yourself. There's just no way.
Right. That's, I think that we always face with going to the core, choosing between flow cytometry and the title. Is time, how gentle the application is toward our nuclei. Nuclei can burst, we can both things clump together, try to run the 10X and just move out of line.
Yep.
So that's one of the things that, the title then had some problems with before chips with something that we can get clogged. So what we're doing is advanced chip technology to make it more reliable mice. We stopped using it. We have to use the flow core, both using the title just because once it clogs, it's tough to recover the cells then.
Yeah, and we can talk too about kind of that sample prep upstream of kind of what you guys are sorting. Because again, any microfluidic instrument, right, there's a channel. So, and there's a 20 micron filter on there. So depending on the size of the cells, kind of what's coming in, that's where we kind of set up a whole kind of sample prep workflow upstream to really optimize that. But if you guys are doing nuclei, I mean, depending on your cell type, we have lots and lots of customers very successfully sorting nuclei on the title very quickly, very efficiently with great results. And actually I have a lot of data here and some testimonials too that I'll share. So we can kind of talk off once you're with the core, Frost can kind of work with you to kind of figuring out maybe why the upstream could potentially have been causing that. But we will definitely help you out with that.
Yeah, the 5,000 what chip you want. Regular speed versus high speed. The good timing.
Yeah, they're both, I mean, for the most part, like both, the difference between the two companies is the speed, like internally they're the same. So I work with David, a lot with a lot of customers that, some samples we had initially like issues that they clog, they stick, and you have to identify what is the issue. Are the cells clogging to each other? Are they, sticking to the filter? Is the buffer not compatible? Is there a lot of cells? And there was a lot of tough examples where we had to maybe troubleshoot 2 or 3 times until we then found the perfect condition for these cells to just, sort perfectly. So it's almost, a lot of, we have a lot of, if the cell is less than 20 micron, we can all work up to you until we find the perfect conditions, buffer, everything, dilution, and so we'll talk after this because I'm happy to come on site and help you, figure out what's leading to that process. We work with Morgan as well to identify maybe upstream with that, how we can help with that. Then we'll,
Good.
Yeah. Awesome. Okay. So again, we're going to be talking about the Tito. For those of you who have not seen it, we'll talk about it here, but we'll also show you in person. We're going to be running a quick Bio2C sample. So the main goal here, right, is prepare cells in single cell suspension. That's obviously the whole point of Morgan's talk, is getting to that nice quality single cell suspension. You're going to transfer your cells to a cartridge right here. This is the heart of the system. You're going to load your sample, draw your gates, and press play. Let's see if this video will play for me. This is just kind of a little video kind of showing the workflow here. You can see the cartridge there. Scan each barcode. So temperature, you go to your cartridge, initiate, it does kind of be aligned with the lasers. You don't need a top plate. You can other kind of things you want to set the software, you select your population, and again, this is your market. So that's okay. So let's go into kind of the actual unit itself. So you'll see it today, but again, that's it, right? A lot of times with other traditional droplet cell sorters, you either see them in a hood because they have to be in a hood, or you see buffer bottles everywhere, you see tanks of fluid. The instrument is really an instrument with optics, obviously the digital processing, but there are no shared fluidics in this instrument, right? So you're not going to have really any kind of extra things on the bench, which is really nice. Again, we have the 3 lasers or 8 fluorescent channels. We also have our scatter channels. Next year, we are coming out with our newest Tito instrument, which is going to have a 4th laser. That's going to be a yellow-green laser. So that's super exciting, along with a lot of amazing updates to the software that have been in the kind of queue for requests for a long time. So we're super excited to get that launched next year. But again, the heart of the instrument is the cartridge, right? This is what makes it so unique and so special when it comes to cell sorting, and unlike kind of any other cell sorter you see on the market. So everything happens in here. There is no tubing in the instrument, right? There is no shared fluidics. When you load your sample stays in here the entirety of the sort, okay? So you have your input chamber, you have your positive collection chamber, and your negative collection chamber. The way the actual mechanics work is that we actually have the world's fastest valve on the bottom of each microchip. This valve can actuate 33,000 times a second, okay? So as a cell kind of passes through, the valve simply opens if it's a cell of interest, allowing it to flow through to positive. Anything else to flow through to negative. Couple things. What that means is that we don't use sheath fluid. So you can kind of sort whatever media or buffer your cells are happiest and most compatible in. We've had some customers do some really cool things. We did actually on here we did sea anemone that were sorted in saltwater buffer because they just died in droplet cell sorters and actually survived and had the correct morphology and functionality post sorting on the Tito. We have people even preload that positive collection chamber with different buffer medias that they want their cells actually sorting into. We've had some really cool kind of different metabolomics things we've done as well with different kind of medias and buffers. But again, there's no sheath fluid. You can sort whatever media buffer they're happiest in. And because we don't have a waste, right? We have a positive and negative. So that means you have accountability for your entire sample throughout the entirety of the sort. So what that means if, a clog or, you realize you messed up for something, you can access your sample, right? So it's either in the positive, the negative, or the collection chamber, okay? And what that also means is that you can do whatever you want with that negative. So whatever you didn't sort, you can resort. You can culture, you can do flow cytometry, right? So there is no waste. You're never having to lose cells. People always ask, wait, the positive collection chamber is only 2 mLs. Isn't that going to overflow? Well, no, this instrument is different. You actually are concentrating your cells. So unlike any other traditional droplet cell sorter, you have a lot of liquid. These cells come out actually a little more concentrated than what they come in, which means that you don't need to do additional spin down steps, which a lot of times you can lose up to 25% of your cell recovery. So a lot of times people can actually start with less amount of sample or cells to get to that downstream number. And again, this isn't going to replace any droplet cell sorter, right? This is not necessarily an apples to apples comparison, but this is going to help, help optimize certain application cell types that might be trickier to get through a droplet cell sorter. So again, kind of just the difference in kind of how the mechanics of the system work. We don't have a charge. There's no decompression. It's extremely low pressure, 1 to 3 PSI on the standard and 5 to 8 PSI in the high speed. No aerosol, so a great option if you guys do any kind of infectious material, BSL-2 plus work. This has been validated by the NIH to be a BSL-3 benchtop cell sorter. So in all of our BSL-3s and even BSL-4s, it's not in a hood. It's actually sitting on the bench. And then again, no aerosols and no cross-contamination. So for a flow core, it's great because if you have people doing all different types of cell types, you don't need to decontaminate the tubes because there are no tubes. You simply have your cartridge and you throw it out. That's the SOP for kind of the decontamination. And again, something that's really powerful for sequencing is, again, that sorting creates that concentrated product, right? So it can be in the desired buffer, which you can often put directly into sequencing. Your sort setup is minimal. Once you kind of learn it, you have your template set up and kind of keep on going back to do that sort. And just showing the difference in the volume and the concentration. And then typically what's recommended for 10X is actually within that range that we actually sort at. So you get that volume and that concentration. So they're ready for downstream applications and you don't need to do any kind of additional steps. So real quick, kind of talking about, testimonials and referrals and stuff like that. So we have a ton of published papers, a ton of collaborations using Tito and genomics. Again, see here. Again, if you guys want to access any of this, I can make sure to send it out to you. Again, nuclei is a huge thing that we do a lot of work with our customers. So this is, we have a poster available for this, but again, kind of using our upstream workflow to do our nuclei sort and just showing that, obviously isolated, no debris and little to no blubbing, which is great. Again, we have all these papers. Again, quick nuclei sort, and this was their standard cartridge, so we can even do it twice as fast now. But really simple sort, high purity, low sort time. Nuclei is actually a great application for the instrument. And this is from Cedars-Sinai's genomics core. So the genomics core itself actually has their own Tito. They use it upstream of every sample, almost as like a sample prep instrument. They do a lot of nuclei. So you can see here, they had great success using the Tito to sort nuclei from various tissue types, extremely user-friendly, and the sorts were quick. They did it in the standard cartridge, so 1 ml in 15 minutes, but the high speed would have been 7.5. With the low pressure, the nuclei remained intact with little blebbing. Post-sort nuclei yields increased by 30 to 35%, allowing them to perform their robust single-cell assay. So again, this is literally in a genomics core where they don't have any flow experience, and they're using it upstream of all their samples. This was actually done here on campus back during COVID by Patrick Wilson's lab. And just kind of a testimonial from him. So they were doing very rare subpopulations of B lymphocytes recovered from COVID-19 patients. Quick, simple. They were able to safely sort their BSL-2 plus materials at the flow core. And again, because of the nature of the Tito, high cell viability, which then enabled them to perform 10X on very rare population of cells. This is not genomics, but metabolomics. This is actually a really big group at Northwestern that is doing the Tito for all of their assays upstream of metabolomics. But Sam Weinberg, so just again, reducing cellular stress, eliminating background contamination, flexible workflow that is ideal for isolation of rare cell populations, powerful instrument for evaluating cell metabolism without, limiting alterations during the isolation and purification process. And then another one here, this is again from Cedars-Sinai. Since the sample sorts into a negative chamber, if any issue arises, it's kind of easy to resort without any sample loss. You kind of just recover it from the negative chamber. And then again, up close and personal right here, David, obviously we love to hear that it's, one of the favorite acquisitions by the facility. Again, offering just kind of new opportunities in the way that, you guys handle the cells that are just, again, not available with the way traditional droplet cell sorters work, right? So, where the quality and viability matters, that's kind of where the Tito kind of takes precedence. And of course, that includes single cell sequencing downstream. And again, it's a really easy platform to use as a user instead of needing to have an expert. So just kind of an overview, right, of kind of the Tito instrument itself. And again, if you're going to the workshop, you'll see it in person, you'll see it sorting. But again, your temperature control, your, high sample quality due to the gentle sorting, It's actually a lot of times just used as like a cleanup step, sample prep step. You can do your aerosol-free, sorts-free infectious material. And again, the sample is always kept in the cartridge. You're never going to have waste. You're never going to have anything that you don't have access to. And then it also highly concentrates itself. Okay, so again, kind of a summary of our portion is the importance of sample prep, right? And the importance of what you're putting in to your downstream analysis is going to really determine the results afterwards, right? So again, using all of our technologies upstream, which has been proven to elevate those results, including, you know, the tidal cell sorter, making it really easy to kind of get to those rare populations, especially tricky sample types, nuclei, a lot of things that are used for 10x. So that is our part. Does anybody have any other questions? Yes.
Can we give suggest this one to us when we were samples? The one question we were stuck at is how to recover the samples post-sort.
Yep. And for us to kind of go over the technique, because again, it's very, you're going to see very small volume, right? I think it's like 0.3 picoliters of volume that actually comes with the cell. That's why it's concentrated, right? So when the valve opens, it's really only taking the cell very small volume. A lot of times people preload that chamber with a buffer or media that they want to sort into to kind of make it easier. But again, I don't know if you guys use those like long gel loading pipette tips, but there's obviously some technique to kind of recovering because it is such low volume. But then you can, you know, obviously add your, you know, buffer and kind of recover it that way. But we can kind of show you, if you're coming to one of the workshops, how to best do that.